Hello everyone, welcome back. Every cell in your body is running a silent race, a race to produce energy. But have you ever wondered what is the very first step that fuels this entire process? Welcome to glycolysis, the pathway where glucose begins its journey of breaking down to release energy. Today, we are not just learning the reactions, we are decoding one of the most fundamental survival mechanisms of life. So, in today's class, we will be discussing about the pathway glycolysis. We will be learning about how to define glycolysis, and we will be describing the steps and enzymes of glycolysis, and we will be explaining the reactions of glycolysis together. Then we will be discussing the energetics of glycolysis. So, glycolysis. This is a word derived from Greek terms. Glykos means sweet. Lysis means splitting. So, in short, glycolysis means the lysis or breaking down or the splitting up of glucose molecules. And this pathway is also known as Embden-Meyerhof pathway. Embden is a scientist who have described the steps for production of lactose from pyruvate, and Meyerhof is a scientist who described the whole reactions and steps in glycolysis pathway. Moving on to the definition, glucose, it is split into 2,3-carbon pyruvate molecules, and this happens under aerobic conditions. And in anaerobic conditions, this pyruvate will be converted to lactate, along with the production of small quantity of energy. So what is the site of reactions? All the reaction steps take place in cytoplasm. Now, what is the significance of this glycolysis pathway? It is the only pathway that is taking place in all cells of human body. And this is the only source of energy in RBCs. In strenuous exercise, when the muscle tissue lacks enough oxygen, anaerobic glycolysis, it forms the major source of energy for muscles. So with this introduction, we are moving on to the different reactions of glycolysis. So we have two phases in glycolysis. The first one is preparatory phase. Second one is splitting phase. Now we are moving on to discuss the preparatory phase of glycolysis, which has three steps, step 1, 2 and 3. Step 1 and 3 is phosphorylation and step 2 is isomerism. So let's see the first step, phosphorylation of glucose. So here, as you can see here, this step is irreversible. That means only the forward reaction is happening. From glucose 6-phosphate molecule is formed. You can see here ATP is required for this particular step, and there is presence of magnesium ions. The enzyme can be hexokinase or glucokinase. Any of these enzymes can catalyze this particular step. So what happens is the phosphate from this compound is added up to the glucose at the 6th position. As you can see here, at the 6th carbon atom, phosphate group is attached. That is why it is known as glucose 6-phosphate. That is step 1, which is irreversible step, and it is catalyzed by either glucokinase or hexokinase. Now let's see what are the differences between glucokinase and hexokinase. So hexokinase, it is present in almost all tissues, and glucokinase only in the liver. And as you can see here, the Km value for hexokinase is low, and for glucokinase it is high. When compared with the hexokinase, the value of Km is high. What is Km value? It is the substrate concentration at half maximal velocity. So Km value will be inversely proportional to affinity. So as you can see here, hexokinase have low Km value, so the affinity towards the substrate will be high. So even when the blood glucose level is very low, the glucose is utilized by body cells. So that is the importance of Km value and affinity of substrate. Now here you can see the glucokinase, the Km value is high, so the affinity towards the substrate will be low. So it acts only when the blood glucose level is more. So if it is more than 100 milligram per deciliter, then the glucose is taken up by the liver cells for glycogen synthesis. So that is the importance of Km value and affinity in case of glucokinase enzyme. And the specificity, it acts on glucose, fructose and mannose, but glucokinase acts only on glucose. The functions we have already discussed. Moving on to the second step, that is isomerism. So as we can see here, the enzyme which is catalyzing this reaction is phosphohexoisomerase, and this is reversible step. Okay, so from the previous step, that is step number 1, will give you glucose 6-phosphate, and using this particular enzyme, glucose 6-phosphate is getting isomerized to fructose 6 phosphate. Fructose 6 phosphate. So that is the step number 2. Now, moving to step number 3 of preparatory phase, that is phosphorylation. The phosphorylation, it is again irreversible step. And here ATP is required and also magnesium is also required. And fructose 6 phosphate, which is obtained from the step number 2, it is getting converted to fructose 1,6 bisphosphate. So as you can see here, so the sixth position, sixth carbon atom, it has phosphate group in fructose 6 phosphate, and the phosphate group from this ATP is donated towards this compound, and then it is getting converted to fructose 1,6 bisphosphate, where at the first position, first carbon atom also has phosphate group. Now the enzyme is phosphofructokinase. And I want to tell one more thing. This is a rate limiting step. That means this particular step is happening in very slow manner. And this particular enzyme is, so the regulatory enzyme, which can regulate the whole glycolysis through mediating this particular step, that is phosphorylation step. Now, moving on to the next phase, that is splitting phase of glycolysis. So step number 4, that is cleavage, and step number 4a, that is isomerism. So step number 4, cleavage. So fructose 1,6 bisphosphate is a compound we have received from the previous step. Now in this step, aldolase enzyme, it splits this 6 carbon compound into 2 3 carbon compound. So as we can see here, dihydroxyacetone phosphate is formed, which is a 3 carbon compound, and glyceraldehyde 3 phosphate is again a 3 carbon compound. So this particular step is reversible. Now, in the next step, we will see how these two compounds are interchanged. So, step number 4a is isomerism, and it is reversible step. Here, triose phosphate isomerase, the enzyme which can isomerize dihydroxyacetone phosphate to glyceraldehyde 3 phosphate. And the reverse also can happen with the same enzyme. So, that is step number 4a, isomerism. Now, moving on to the next step, that is oxidative phosphorylation. Oxidative phosphorylation. So before moving on to the step, I just want to add a few important points regarding oxidative phosphorylation. Here, the energy is trapped by oxidation of reducing equivalents. That is just like NADH. So energy is trapped by oxidation of reducing equivalents in oxidative phosphorylation. So here you can see the glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate is acted upon by glyceraldehyde 3 phosphate dehydrogenase, and this is NADH dependent step. So here there is addition of one phosphate molecule also, and this phosphate is getting attached to this particular position like this. So the third carbon atom, it already has phosphate group, and the first carbon atom, here also there is addition of phosphate group, and it forms 1,3-bisphosphoglycerate molecule, and as you can see here, this is a high energy compound because of the presence of high energy bond. Clear? Now, next is substrate level phosphorylation. So again, before moving on to the explanation of this particular step, I just want to explain what is substrate level phosphorylation. Here, the energy required for this reaction, it is trapped from the substrate. As we can see here, this is the substrate of this reaction, right? And energy required for this particular reaction is taken up from the substrate compound. As you can see here, there is a high energy bond. So substrate level phosphorylation means the energy is trapped directly from the substrate. So as you can see here, with the help of bisphosphoglycerate kinase, ATP generation is happening here, and here this phosphate group is taken up into the ADP molecule, and then ADP is getting converted to ATP, and the resultant compound, the product of the reaction, doesn't have the high energy compound, and that is known as 3-phosphoglycerate. Because the first position, there is loss of phosphate group. Now it becomes 3-phosphoglycerate molecule. Now this I have already explained. I just want to tell for substrate level phosphorylation, step number 9 is also an example that we will be discussing in the coming slides. Step number 7 and 8. So here, step number 7, it is just the isomerism. 3-phosphoglycerate is getting converted to 2-phosphoglycerate. So here you can see these are the 3 carbon atoms, and this compound, it has phosphate group at the 3rd position, and it is getting changed into the 2nd position, and this is known as 2-phosphoglycerate, and the enzyme which is catalyzing this reaction is phosphoglycerate mutase. Now moving on to the next step, that is step number 8, which is catalyzed by enolase enzyme, and this particular enzyme, it has removal of H2O. As you can see here, H2O is removed from 2-phosphoglycerate, and this is getting converted to phosphoenol pyruvate, which is again a high energy compound, phosphoenol pyruvate, and which is catalyzed by enolase enzyme. So I just want to add one important point regarding the step number 8, why we are using fluoride bottles for collecting blood samples for glucose. So fluoride, it will reversibly inhibit the enolase enzyme. So what happens? The fluoride will stop the whole glycolysis. So when we are taking blood for blood glucose estimation, we have to use fluoride bottles. If you are not using the fluoride bottles, the glucose, it is metabolized by the blood cells. So when we are measuring the blood glucose, lower values of blood glucose values will be obtained for that particular patient. That is why we are using fluoride containing bottles, that is grade top blood collection tube. Moving on to the step number 9, it is again a example for substrate level phosphorylation, and here you can see there is production of ATP, and this is the substrate level. So the energy is trapped from the substrate compound, and it is getting attached to ADP, and which is converted to ATP. And this particular step also requires magnesium. So from phosphoenol pyruvate, with the help of enzyme pyruvate kinase, pyruvate is formed. So this is the step number 9. So till this particular step, it is aerobic glycolysis. Okay, now what happens in anaerobic conditions? In anaerobic condition, this pyruvate, it is getting converted to lactate with presence of lactate dehydrogenase enzyme, and there is involvement of this particular coenzyme NADH. So this happens only in the anaerobic conditions. Pyruvate is getting converted to lactate. So in short, what happens is, so from this is the step number 5, that is glyceraldehyde 3-phosphate, and there is production of NADH, right? And this particular NADH is taken up in the step number 10. Step number 10 happens only in anaerobic conditions. Anaerobic conditions. So this NADH which is formed in the step number 5 is utilized in the step number 10, and there is formation of NAD+, which is again used up in this particular step number 5. And if there is lack of oxygen, the pyruvate conversion is not happening. Pyruvate to acetyl-CoA is not happening. That is why pyruvate is channeled to lactate formation. Right. Now, in the whole glycolysis, what are the irreversible steps we have studied? Step number 1, 3, and 9. And ATP requiring steps are step number 1 and 3. ATP generating steps are step number 6 and 9. And NADH generating step is step number 5. And 1 NADH is equivalent to 2.5 ATPs. Now, let's see what is the energetics behind glycolysis pathway. So first, we will be discussing what happens in aerobic conditions. So the first steps, that is step number 1 and 3, there is utilization of ATP. That means ATP is utilized. So 2 molecules of ATP is utilized. So that is altogether, that is -2. Now, the step number 5. Step number 5, here there is production of 2 molecules of NADH. Why? Because when we have studied about the step number 5, there is generation of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And this dihydroxyacetone phosphate can be converted to glyceraldehyde 3-phosphate. So in short, from one particular glucose molecules, 2 molecules of glyceraldehyde 3-phosphate is formed. That is why this particular NADH, 2 molecules of NADH is formed. Okay, so here 1 NADH means 2.5 ATPs. So 2 NADH means it is 5 ATPs. Now the step number 6 and step number 9. So after the step number 5, we are discussing about 2 molecules in each step. 2 molecules of glyceraldehyde 3-phosphate. So in step number 6, there is generation of ATP. So that means 2 molecules of 2 molecules of ATP is formed. So again, it will be +2. And in step number 9 also, there is generation of ATP, that will again count as +2. So altogether, there is 9 - 2, 7 ATPs. So in aerobic conditions of glycolysis will give you 7 ATPs. Now, what is the change we are expecting in anaerobic glycolysis? In anaerobic glycolysis, the hexokinase and phosphofructokinase, there is requirement of ATP. So, that is -2. And the NADH, which is generated in the step number 5, is utilized in the step number 10. So there is no net generation of NADH because whatever NADH is produced in this particular step number 5 is utilized in step number 10. So that's why we are not counting NADH here. Now, step number 6 and step number 9, there is generation of ATP. Since we are counting it as 2 molecules of glyceraldehyde 3-phosphate, we are counting plus 2 ATPs in the step number 6 and plus 2 ATPs in step number 9. So that means 4 minus 2 will give you 2 molecules of ATPs. So in anaerobic conditions, there is generation of 2 molecules of ATPs. So I hope you have understood whatever is taught in this particular session. Thank you for listening.